Thank you. Thank you for coming back so promptly. I know that the day has given us lots and lots of food for thought and lots of opportunities for discussion. And that kind of neatly brings us into the people that we've been trying to influence and engage in conversation. So it gives me real pleasure to introduce the afternoon session around the views from our regulator colleagues. And I'd like to invite Susan Winckler on the stage from the Reagan Udall Foundation for the FDA in the USA. Thank you, Susan.
Well, it's great to be here, and I thank you for the invitation and to join. So let me do a little bit, actually, Dr. Muenzer and I were just noting, neither he nor I are regulators, but we're on the panel. But I am distinctly regulatory adjacent, and so let me explain a bit. The Reagan Udall Foundation for the FDA was created by the Congress in the United States. It was created with the belief that a regulator, in our case, the Food and Drug Administration, would be more efficient, effective, and transparent if there was an opportunity to routinely engage with the outside world, or as we say in the U.S., with an email address that's other than .gov. So to have that opportunity to interact, and so that's why our foundation exists. And so about a year ago, we held a meeting that was similar to the incredible discussion that we've been having today. It was slightly different in that our approach in the one-day meeting was intentionally at a higher level to say, let's talk about biomarkers in rare disease and the importance of biomarkers in rare disease, with the heparan sulfate in neuronopathic mucopolysaccharidosis as a case study for that. So a slightly different orientation. Another, I think, just important difference, because our foundation is regulatory adjacent, everything that we do is then coordinated with the FDA to assure that we are helping them do more to protect and promote the public's health. So we constructed the meeting with input from the FDA as well as from the non-government side to assure that we could explore various topics together. And so that the structure is reflected here on the slide, generously supported by a couple of the companies. And then they created enduring material, and that there's a workshop summary that's available on our website. So if we think about why we gathered, the intent was not regulatory decision-making, but rather collaborative learning and sharing. So the sharing from experts in the private sector and those in government to better understand each other and the important questions. So we tried to be sure to address these three different things, to talk about broadly the role of biomarkers in rare disease, where everyone in this room and online today has gotten an excellent immersion into that connection and why it is that we need something other than just solely a clinical endpoint. To also understand the U.S. in the U.S. FDA structure, the accelerated approval pathway, which has that connection between a biomarker that's reasonably likely to predict clinical benefit and then applying that in rare disease and bringing innovations to market on something short of what we might expect to see through the traditional pathway. Although importantly, even when using the accelerated approval pathway, then there's research that's required afterwards to assure that that clinical benefit is achieved. And then the third premise was to look at this through the eyes of heparan sulfate. So with that, then what would success look like? We decided that success in this space for that one-day meeting would look like something like a joint scientific exploration or a continuing medical education meeting where you had everyone learning and testing questions from various perspectives. So if we think about, we just wanted to describe the meeting a bit more in saying from a speaker perspective, that meant that the audience heard from the FDA itself, from those in academia and researchers, from the companies, and as importantly as we heard today, from the patient representatives, so that you're getting that full circle of who is involved and affected by these decisions. That was similarly then reflected in the attendance that also similar to this structure, had a number of individuals who were in person and then a much broader audience in the virtual world to see and hear. But similarly, we had a number of regulators who were listening, industry to understand what is the target and what it is that we're pursuing, and then also from the patient perspective and from others who are in the stakeholder community for developing medical products. So I'd close with the take-home messages, and I'm thrilled that it's one thing for me to say what I think people heard because I spent my day moderating the meeting. Dr. Muenzer will pick up with what an actual participant and contributor heard, which is far more valuable than what I perceived. But what we took away is that these types of collaborative efforts can help drive innovation, where you can learn what has worked in one environment and, just as importantly, what hasn't been, to better understand important questions about the analytical validation and having a clear picture of the accuracy and precision of the biomarker measurement. And I think as powerfully as we've felt it in this room, 13 months ago it was similar, the importance of the patient centricity and why this work is done. That is always a powerful reminder and grounding for all of those different sectors who are involved in this regulatory activity. So that was a recap of what we did just over a year ago, and I'll turn it back to our moderator because I know exactly how important that is. And then Dr. Muenzer can tell you what he heard, which is a far less biased perspective than what I hope we facilitated. Thank you.
Thanks so much, Suzanne. Dr. Muenzer, I'd love you to share with our audience the overview and the outcomes of the workshop.
Again, while we're waiting for the slides, it's certainly my pleasure to be here, a topic that I think is so important. From an academic perspective, again, I'm not a regulator at all. I'm an academic person. I hold a distinguished chair. I'm also, as you heard, the director of a recently, last couple of years, MPS Research and Treatment Center, which has my name on it, which is really sort of, to me, a unique honor based on a donor. I have disclosures involved in pharmaceutical companies and involved in current clinical trials. You've heard from Susan. This meeting was unique. It really brought together academic physicians, scientists, patient advocacy, and industry, and the FDA all in one place to hear about all the unique features that we've heard today already in terms of neuronopathic MPSs. I want to just highlight these are ultra-rare genetic disorders. In the US, there's probably no more than 2,500 total MPS patients, but for MPS 1, 2, and 3A, there's probably less than 500 patients of all different ranges of severity. Again, the biochemical hallmark is urine glycosamine glycans, which are really the primary defect, as we heard, and it's really that interlysosomal accumulation of GAGs that causes secondary lysosomal dysfunction and then secondary cell death and all sorts of other toxicities. From my perspective, you've heard this, but I think it's important to summarize for everybody. These are very heterogeneous disorders. They're rare, they're progressive, and they're clinically characterized by both physical and central nervous involvement, And unfortunately, most patients die prematurely. And sometimes the untreated historically Hurler patients, the severe MPS I, died less than 10 years of age. They appear normal initially, but develop both somatic and cognitive impairment. You've already heard that. And again, I won't emphasize, but the severe form of MPS I, Hurler syndrome, 2, 7, and all Sanfilippo's all develop cognitive impairment. So what are the clinical trial challenges from my perspective that came out of this, from this regulatory meeting? Again, except for the severe 7 patients who develop, can present at birth with, non-immune high drops. Typically, most individuals will develop normally for the first 1 or 2 years, start to slow down, plateau, and regress. But the challenge is the rate at which they either slow down or regress is variable. How long the plateau is variable, so it makes it really challenging from a clinical trials perspective because of that. We already heard today, I want to emphasize, the clinically diagnosed MPS II patient with neuronopathic disease, as far as I'm concerned, has irreversible brain disease at the time of diagnosis. Their markers will go down, but if you take that patient 3 or 4 years later, they may not get as well as you want because they already have impairment when we start from the clinically diagnosed patient. Again, hopefully in the future in the US, we'll have newborn screening for some of these disorders, and we'll be able to do a much better job of picking up early and really preventing. But right now, it's really sort of stabilizing. Again, replacement of the missing enzyme in the brain of a neurocognitive individual with cognitive impairment will not result in cognitive improvement from my perspective. At best, you're going to stabilize them at best, depending on where they are. In some places in the disease course, you're not going to even stabilize them. The disease is going to keep progressing at a slower rate. So from my MPS clinical experience, 45 years of doing this, over 600 to 700 MPS patients. That's sort of my perspective. To attain a statistical significant difference, which is what the FDA wants. They want that p-value of less than 0.05 in a placebo-controlled trial. If you take individuals in the trial all younger than 3 years of age, you at least have to have 3 or 4 years duration of trial, if not more. That's the challenge. And again, the control group, what's going to happen to them? They're going to develop progressive, irreversible brain disease. From my mind, for the regulatory bodies to require companies to do placebo-controlled trials, it's really unethical. The companies are in a bind. Historically, prior to the Reagan Udall meeting, they were forced to do placebo-controlled trials, otherwise they would not get their product registered. And again, utilizing CSF heparan sulfate, an accelerated approval pathway, is really the logical long-term follow-up that has to be done for this population. So why CSF heparan sulfate is a reasonable likely to predict clinical benefit? And I'll just summarize that point. One point hasn't been made yet today. Lysosomal enzymes are only active in the lysosome in the cell. They have virtually no function at a pH of 7.4 in your serum. So just because you see a serum enzyme elevation, that doesn't mean that enzyme can do anything for lowering GAGs. Yes, it's important there if it goes in cells, but if in the serum. Again, heparan sulfate is the primary marker. They correlate with brain tissues. There's lots of data, animal data, and we saw already today here how CSF heparan sulfate correlates with brain tissue. The other point that hasn't been made is that CSF is a dynamic fluid. It turns over 3 or 4 times a day, if not more, and therefore if you see a change, that reflects what came out of the brain cells. And again, so the only way CSF heparan sulfate can be reduced is that if you reduce brain tissue heparan sulfate. And then again, lowering is certainly a reasonable likelihood to predict clinical benefit. But again, it depends when you start. If you start with already significant cognitive impairment, you're not going to get any better. And as we already heard, regulators like clinical improvement, but no improvement in this progressive disorder is clearly benefit. One of the things I did at the Reagan Udall Foundation is give my 10 reasons why heparan sulfate using the FDA accelerated approval pathway should be utilized in neuronopathic MPS disorders. Incredibly rare disorders. You cannot do, I think Carol talked about 1,000 patients. When I did the phase 2-3 clinical trial for Elaprase, we used half of the eligible patients for that trial. We had a one-time chance to get it right. And that's one of the challenges. The biochemistry is clearly understood and known for 20 to 30 years. We know the primary event, CSF heparan sulfate, is always elevated in neuronopathic MPS patients, always elevated. It can now be reliably measured, as you heard from Fritz earlier. 10 years ago, that was more challenging. Today, we have different assays, all are reliably can be measured. As Fritz already alluded to, companies need to stake one assay and continue that same assay for that development. I think they're comparable. Again, CSF heparan sulfate correlates with brain tissue heparan sulfate. When brain tissue heparan sulfate goes down in animal models, CSF heparan sulfate goes down. Reduction of secondary markers is also associated with we see GM2, GM3, and you heard about NFL, all support the relevance of CSF heparan sulfate as the primary biomarker. The reliance on clinical efficacy with the placebo-controlled trials to demonstrate effectiveness, to me, is unethical in children with slowly progressive, irreversible brain disease. It's just not, Simon talked about we need a new design. Well, the design is that placebo-controlled trials cannot be utilized successfully in this disorder. We need to be some other mechanism. And again, we need this regulatory flexibility. And clearly the Reagan Udall Foundation gave the FDA more impetus. And maybe it was one giant CME, continuing medical education, but it clearly gave the FDA what they needed. So summary from the Reagan Udall, just very briefly. In MPS animal models, CSF levels correlate with brain tissue heparan sulfate, and in turn, pathological and clinical outcomes. There was very nice data that was presented. CSF heparan sulfate is a predictor of clinical outcome and downstream biomarkers of lysosomal and neuronal function. Substantial data was presented with CSF heparan sulfate is a valid biomarker for neuronopathic MPS disorders. And again, the accelerated approval pathway is really the way to go to really allow much more timely, efficient approval of these rare disorders that life-saving drugs. The unique thing about this, there was a paradigm shift. We've talked to the companies involved in the FDA after the Reagan Udall Foundation, before where they heard no, no, no. Now they're saying we're willing to talk about accelerated approval. 3 companies in 2024, each were given the approval to file their BLA or their biological license application using the accelerated approval pathway for both MPS 2 and MPS 3A. You've heard already Ultragenyx has that completed. They have a so-called PDUFA date where the FDA's got to give them an opinion in 6 months in August. So that's going to be fantastic. Again, they are using, this is gene therapy, as you heard from San Filippo, and they're using non-reducing ends as their biomarker. Regenix just recently completed the rolling BLA, so that's already completed now for their gene therapy, as you heard for RGX-121 for severe Hunter syndrome. They're using CSF D2S5, a specific disaccharide, as part of the accelerated approval pathway. And then Denali, as you also heard from Carol this afternoon, expected to submit a BLA for DNL310, again, under the accelerated approval pathway, using using heparan sulfate based on total disaccharides for their IV ERT designed to cross the blood-brain barrier in early 2025. So hopefully we'll hear them very soon in terms of that announcement. Thank you.
Thank you so much, Joe, and that gives us a real insight into the opportunities that regulators can consider, which gives me great pleasure now to invite Peter Marks. And Peter's not here in person, he's going to be presenting online, and he's going to talk a little bit about facilitating that accelerated approval. Peter, can you hear us?
I can. Can you hear me okay?
We can. Welcome. I'll hand
Great.
you over.
Okay, thank you very much. So I think this will pick up off of Dr. Muenzer. First of all, I think the concept of using accelerated approval to help move these products forward um, is nothing new to us at the Center for Biologics, and it's something that we have been thinking about for years before the Reagan Udall workshop. And I think it was very helpful because it helped focus us, that workshop helped focus us on this particular heparan sulfate biomarker. And the next slide, please. You know, I think one of the impressive things is we now have a number of gene therapies approved in the United States, 22. Some are modified T cells or stem cells, and several of them are directly administered. And we recognize that the only way we're going to get to having a larger number of these gene therapies is by leveraging our accelerated approval pathway, in part because otherwise the development programs just take too long. Go to the next slide. When you look at the key challenges here, we could, and we'll save the manufacturing challenges for another day, and we can talk about different global regulatory requirements some other time. But clinical development timelines are what cause companies to run out of cash and lose interest in developing products, as is very well illustrated in the MPS field. Next slide, please. If you look, biomarkers in this area, and I would say, although we're going to use MPS as an example, you know, these are really important in the world of gene therapy because they complete out something that is very helpful here. In gene therapy, since we know the intervention that we're making, we know the pathway that the intervention is affecting, the biomarker in a sense completes out or helps complete out the scientific data that helps us understand that we may be likely to predict clinical benefit. So it really is something that plays a critical role in facilitating how we apply the accelerated approval pathway. And I think, you know, in some cases, it's directly connected to the therapeutic intervention. For instance, sometimes the gene that you're inserting is actually what you can measure, and understanding that activity, such as in hemophilia, allows you to pretty much directly predict clinical benefit. Alternatively, sometimes you have to do it indirectly, such as we're doing in the case with heparan sulfate in MPS. Now, that I think it goes back also there kind of interestingly to the fact that if one actually takes it established that heparan sulfate is the toxic metabolite here, or is one of the toxic metabolites, one could argue that you have an important measure as well. But as we think about these, one of the important things to also recognize for us at FDA is that accuracy and precision of measurements are critical. If I have the next slide. I think that's something that also was touched on earlier today, that, you know, accuracy, basically referring to the closeness of a measured value to a standard or known value, is something that we need to feel comfortable with because it correlates with clinical truth. If I have the next slide, precision or being able to measure something reproducibly is also something that's important to us, because if you want to see changes over time, you have to have assays that are reproducible over time. And I think what was presented earlier today is that we're now at a place where with you know, HPLC and mass spec methodologies, we have the ability here to do this for a variety of rare diseases, but in this case, we'll focus on MPS, where with good accuracy and precision, we can measure a biomarker and feel confident in it. If the next slide, and that allows us to essentially have valid assays which can be applied in drug development programs, which is, again, important for us in feeling comfortable in moving forward with an accelerated approval. In the next slide, please. The leveraging accelerated approval is basically something that, particularly in the gene therapy world, I think this idea, you've heard it mentioned already, this reasonably likely to predict clinical outcomes, it's something that can help us really move things forward in a way that allows us to intervene at a point when children have not suffered irreversible damage or as much irreversible damage as they would if we had to wait for typical clinical endpoints of how someone feels, functions, or survives. That doesn't mean that we'll ignore ultimately getting data on how someone feels, functions, or survives, because we don't collect that. Payers will ultimately lose faith in what we're doing here. But that said, the connection, if it's well made, can save years, because if we can approve something on the basis of a biomarker in 6 months or a year of data on a population, we can potentially get the product to them several years sooner than if we had to wait for a traditional clinical endpoint, particularly because you haven't heard me mention at all that we're talking about randomized clinical trials. Because we're not talking about, we're trying in the gene therapy world to avoid the need for randomized controlled trials whenever we can. Now, in some cases, we can't, but to the extent that we can avoid them, because we have large changes in outcomes with biomarkers that can be measured reproducibly, I think that's something we'd like to try to do. If I have the next slide. And you know, in our world, we're seeing this applied more and more, because for gene therapies, we either have animal models or human observations where you can see that at a given level of either an enzyme or other protein product, that the animals or the humans do not have an observable phenotype, or they have a minimal adverse phenotype. And the idea there is, if you can achieve that level with a gene therapy, you have something that's reasonably likely to predict that you're going to have a positive outcome. That is yet again part of the confirmation here that goes, because you may have more than one marker that you can measure. You can potentially measure an enzyme level plus a biomarker in some circumstances, not in all. But we really feel like this is a way of trying to lean into product development as much as we can. And it's really the only way that we're going to be able to get through the hundreds of rare genetic diseases in a timely manner, because many of them have clinical courses that are so slow that it would take 3-5 years to get clinical data, and you could get biomarker data in 6 months to a year. That's the difference between commercial viability and commercial failure in some cases, or in a fair number of cases. To the next slide, please. So we will continue to push forward on this. We're also trying to do other things. I'll just say a few other words that, you know, we're trying to help small companies move development forward more rapidly. We have a pilot program that is currently ongoing called the START pilot. The idea here is to leverage off of what we learned during the COVID-19 pandemic about enhanced communication with product developers, breaking down the structure of formal meetings, which are scheduled 30 to 75 days out, and having essentially constant communications. We now have a pilot in our center of 4 gene therapies or genetic therapies that can potentially help patients, pediatric patients with pretty severe rare diseases. Next slide. The idea here is, if we can, through enhanced communication, make a difference here, and speed up the development, we might expand this to to become more common in use with rare diseases, particularly in the pediatric setting, because again, months matter. So you can see we picked a variety of different size populations, but in general, what these diseases have in common is that they have serious outcomes in children or young adults. Have the next slide, please. So one other thing, just to touch on this issue of different global regulatory requirements, is that we are trying to work with our partners at EMA and others to see if we can try to speed up development by unifying our requirements so that we don't have manufacturers that have to do rework when they submit to different regulatory organizations. Hopefully, this pilot will ultimately result in us being able to do collaborative reviews, which would again try to speed up development. Have the next slide. So I think we are essentially all in with the idea of trying to use biomarkers as much as we can to facilitate accelerated approval. We have to do so thoughtfully. We have to have do the science well, so that there are compelling connections that are made. I think the mucopolysaccharidoses and heparin sulfate, good example of the science has been done well. Assays now are done well. I think we have the connection done well, and so I think we have a good foundation moving forward. So look forward, and thanks for having me today.
Thank you so much, Peter. And we know that it's some unearthly hour for you this time, so very grateful for your time. It gives me great pleasure to ask Emmely de Vries to join us from the EMA, and she's going to present the EMA's position.
Yes, thank you very much for having me. I realize, having heard everything today so far, that probably it's not a message that is the ideal message or the most optimal message for you to hear, but I do hope I can set something straight and also maybe add some positive notes from my perspective. So what's this one? Yeah. So I work for the Dutch agency, the MEB. And we are, as agency, as the Netherlands, represented in the CHMP, in which we join forces with all the other European agencies. And in the CHMP, this is the committee where the scientific discussions are held. So it will be the CHMP perspective that I am representing here today. So this has also been said before, the reason why we regulate. And we do not exist to be difficult on purpose. We exist to make sure that there are safe and effective therapies of good quality available for patients in Europe. And I think this is very important to stress again. So the basis of our assessment and the basis of an opinion for the CHMP is the benefit-risk assessment. So do the benefits of a product outweigh its risks? And there are a variety of factors that we weigh into this benefit-risk assessment. So, of course, the burden of the disease is very important. We will accept far more risk when we are talking about a life-threatening disease as MPSs than we would do for a headache. We would weigh in whether we are talking about a symptomatic treatment or a curative treatment, whether the treatment would cover all symptoms of a disease or only part of it, for example, the peripheral versus the CNS symptoms. We look at the clinical relevance of the found effect and of the safety profile, also important. And although in principle we make an absolute benefit-risk assessment for the product at hand, we do look at the other available treatment options to make sure that we weigh in the unmet medical need appropriately. So in order for us to make a benefit-risk assessment, it's absolutely essential to obtain a measurable clinical benefit. So I will quickly go over this, not want to spend too much time on this, but there are different types of marketing authorizations that we have in the EU. First of all, the full marketing authorization, where the data package that is submitted by the applicant is comprehensive. We can make a benefit risk assessment. Obviously, this has to be positive, and there are always remaining uncertainties, but these remaining uncertainties do not significantly impact the benefit risk. We also have a conditional marketing authorization or a marketing authorization under exceptional circumstances when the data package that is submitted is not comprehensive. And then it depends whether the applicant will be able to provide comprehensive data within, well, reasonable time frame. And this is not like an absolute value, but comprehensive data will have to be submitted. Or if it's not possible to submit comprehensive data because the disease is very rare, or it's because it's unethical to conduct trials, for example. But very important for both of these types of marketing authorizations, we have to be convinced at the time of marketing authorization that the positive benefit risk is there for the patient. So for both conditional marketing authorization and marketing authorization under exceptional circumstances, there will be specific obligations to address the remaining uncertainties. And you could think about a post-authorization safety study or a post-authorization efficacy study. And for example, these could be registry-based studies where a company is collaborating with an existing disease registry, or they conduct their own observational study. And of course, there's always routine pharmacovigilance, so that's always there. And once, in the case of a conditional marketing authorization, the specific obligation is fulfilled and the remaining uncertainty is addressed, then the marketing authorization will be converted to a full. So to make the switch to biomarkers, because that's why we're here, obviously, today. So there are a lot of different types of biomarkers, and I think this is important to give attention to. If we are looking at pharmacodynamic or response biomarkers, these kind of biomarkers are important to provide confirmation of the mechanism of action of the drug, and they will provide supportive evidence of efficacy. So in this case, the decreased enzyme substrate with enzyme replacement therapy, for example. However, this is not necessarily the same as a biomarker that can be used to address the disease status. For this, for example, although I realize it's also not a validated surrogate endpoint, you could look at the neurofilament levels as a biomarker reflecting the neuronal damage. And then there's other biomarkers, which I'm not going to go into. So from a regulatory perspective, true surrogacy is a very high hurdle. And I'm going to explain to you why. These are different scenarios. And in scenario A, you can see that a certain disease might have a certain effect on the surrogate biomarker, whereas via a different mechanism, it has effect on clinical outcome parameters. In scenario B, we would have an intervention, a drug that addresses the surrogate endpoint, and via that surrogate endpoint might also influence the clinical outcome, but there are different mechanisms additionally to this route via the surrogate marker, which also affect clinical outcome. And in that way, you are only looking at a part of the effect. In scenario C, we would have an intervention that will have an impact on the clinical outcome regardless or independently of the surrogate biomarker. And that will also not enable you to capture the true effect. And worst case scenario, all three of those are at play. So for a true surrogate to be, for a biomarker to be validated as a true surrogate endpoint, you would have to have a situation where the intervention affects your surrogate endpoint and by that surrogate biomarker affects your clinical outcome. And that is difficult to achieve. So the current regulatory challenges for MPSs is that, as you are all very much aware of, there is no biomarker at the moment for which true surrogacy has been established, including heparan sulfate in the CSF. So that makes it impossible for us to translate a given between group difference on the biomarker into a measurable clinical benefit. So we do not know what the sensitivity to change of the available biomarkers is. And also, there is no minimal clinically important difference described of the biomarker, which enables us to translate the difference observed on the biomarker into a clinically important effect for the patient. So, having heard everything that we've seen today, it is really clear that this is a true challenge, and the data that has been presented I think pointed this out very clearly. We do not know what kind of decrease on heparan sulfate we would need in order for the neurons to not die. Or on the other hand, what kind of threshold you will need to reach for the neurons to not die. We can also not really correlate, as was also very clearly shown, the decrease in heparan sulfate with expected clinical benefit, because, as was pointed out, this is related to the stage of the disease. So whereas a normalization of your heparan sulfate in a patient early in the disease is maybe correlated to a 10-20 point improvement on a neurocognitive scale, in a patient that has already progressed neuronal damage, you will have the same decrease maybe in heparan sulfate in the CSF, but there will be no increase anymore on the neurocognitive scale. So I think that really clearly points out the challenges. So the current CHMP viewpoint on the study design that would be necessary is that the randomized controlled trial is necessary to robustly estimate the treatment effect. We've got a very heterogeneous disease, as has been pointed out as well, and it also depends, as we saw clearly from the curves of the cognitive development, where you are in the disease, what is the expected increase, stabilization or decline in that stage of the disease. So we consider that a clinical endpoint is necessary as a primary endpoint. And this will likely mean that 2-year follow-up data is the minimum that's necessary to draw any meaningful conclusions on the cognitive and adaptive behavioral endpoints. That doesn't mean that the biomarker is not important. This should be a co-primary or important secondary endpoint, and the study should really also be powered on it to confirm the mechanism of action of the drug and the pharmacodynamic effect. But I think this is really the most important slide, and this is something that I hope will provide also a positive note to this story. The benefit risk assessment that we do is based on an assessment of the consistency of effects across endpoints. And this is especially important when an appropriately powered study is not possible in rare diseases, for example. So in any case, a consistent effect across clinical endpoints has more weight than a statistically significant effect on the biomarker. When there is consistency across multiple clinical endpoints, personally, I don't care about the 0.05. Statistical significance is not as important as clinical relevance. And do we see, can you convince us that a patient is benefiting from the treatment? And that has, in my opinion, nothing to do with statistics, to be honest. So I was struggling a bit to find you some examples, because obviously we see a lot of dossiers, and that's a lot of data that comes across our desks that we are not able to share. But obviously the reason why we are sometimes hesitant, or often hesitant, on biomarkers is because we've got experience that it didn't work out. So I tried to find some examples as well for you to illustrate the problems that we can run into if we approve too early. So this was Ocaliva, which was registered for primary biliary cholangitis, which is a slowly progressive liver disease. And this company received a conditional marketing authorization based on improvements in liver function parameters, so bilirubin, alkaline phosphatase, AST, ALT. and they had to confirm the clinical benefit in a post-marketing RCT. So this study failed, although they reached quite a high number of the events necessary for them to establish a difference. There was absolutely no difference between the placebo and the treated group on survival, on the liver transplantations that these patients often need. So that started a very tedious process, and in the end, after a referral at CHMP, the benefit risk was negative and the marketing authorization was revoked. And this is obviously for no one a good situation, also not for us, but definitely not for the patients. So to have a different example, this is a drug that was approved for the treatment of phenylketonuria, where the primary endpoint of the study was a pharmacodynamic endpoint, the decrease of phenylalanine in the blood. And that was studied in an 8-week withdrawal, randomized withdrawal part of the study. And the company showed that the drug was able to decrease the amount of phenylalanine in the blood. But this by itself was not sufficient to convince us that the patient was really benefiting from the treatment. And in this case, it's even more complicated because this was a proposed indication for adults. And there's actually not really consensus among physicians what your treatment target should be in adults. So that also made it very difficult for us to confirm what would be a clinically beneficial effect for patients. However, because we saw on the totality of secondary clinical endpoints, we saw a positive trend towards improvement. Patients could eat more protein, were able to release a bit of their diet, which is very strict. There were some trends towards improvement also in the long-term extension study on neurocognitive skills, and this in the end led to us giving a positive opinion of this product. So, as some take-home messages for you today, we need robust evidence of clinical benefit in order to make the benefit-risk assessment. At the moment, in the view of the CHMP, heparan sulfate in CSF alone cannot provide sole evidence for clinical benefit. However, as I've said to you before, totality of evidence is really important here. And I also want to stress that early interaction is key and is really beneficial. We do not bite. We are happy to engage. We are happy to discuss. We are happy to provide our perspectives on any plans that you have. And this can be via national scientific advice, for example, with us at our agency, or via the central scientific advice from EMA. And there's also, as you might be aware, the PRIME scheme, where for products for diseases with a very high unmet medical need that show promise, you can enroll in the PRIME scheme and you have benefits there. So thank you very much. Leave it at that. Thank you.
Thanks so much, Emmely, and I'm sure that has provoked a number of questions, so please make sure that you submit your questions for the panel afterwards. This gives me great pleasure to invite Shirley Hopper on the stage from the MHRA, and Shirley, like Emmely, will give a position from the UK. Thank you, Shirley.
Thank you very much, and thanks very much for inviting me to this meeting. It's been a very interesting and thought-provoking day. Okay, so this is us. So we're the UK medicines and also the devices regulator. And similar to my regulatory colleagues, we're responsible for making sure that these products do meet our standards of safety, quality, and efficacy. So these are some of our responsibilities. Perhaps the bottom right-hand one is that we do wish to collaborate with partners in the UK and also internationally to support earliest access possible to safe and effective medicines and also medical devices ultimately for patients. Okay, so I'm just going to go over our regulatory framework for rare disease and then talk a bit about the different types of endpoints, including those with less evidence and how we might view that. So I'm not going to talk specifically about MPS today. I'm going to talk a bit about assay validation and also clinical trial authorization, and then encourage you to come and talk to us. Okay, so these are some of the provisions, particularly for rare disease. So like EMA, like FDA, we have orphan medicine provisions. So these are incentives for developers to help them to recoup some of their investment in developing these medicines for rare and very rare diseases. Different to EMA and FDA. Currently, we award this at the time of marketing authorization, but we do offer a 10-year period of market protection against similar medicines, an additional 2 years in pediatrics, and there are some reductions in fees. So similar to EMA, we have the conditional marketing authorization. I won't go into details. It was copied and pasted across at the time of EU exit, and we haven't changed it very much. But just to obviously stress that it is for serious or life-threatening diseases where there's high unmet need and where comprehensive clinical data is not yet available. And so we do have a higher tolerance for uncertainty in the benefits and the risk, but overall we would still need to conclude a positive benefit risk. And then, as Emmely said, under exceptional circumstances is for those situations such as very rare disease where it just simply is not feasible to generate a comprehensive data package as confirmatory data. We also have the Early Access to Medicine scheme. So this is to actually to give patients access to unlicensed medicines that might be in earlier clinical development. So also for serious and life-threatening conditions. And we would give a scientific opinion on whether this unlicensed medicine could be used in UK patients, but just to note that that is a free of charge scheme. So the developer pays, essentially. I've gone too far. Yeah. Okay, so obviously the traditional approach to efficacy endpoints is to have a clinically meaningful endpoint. So I'll sort of go through the traditional approach first. But obviously it's worth pointing out that, under our law, to grant a marketing authorization, we do need to be satisfied of therapeutic efficacy and that the benefits outweigh the risks in the target population. So traditionally, the main evidence does come from a pivotal trial where the primary endpoint is clinically meaningful for patients. So obviously things like overall survival, disability progression, and important symptoms. And then if a surrogate endpoint is to be used, such as a biomarker, really it's substituting for that clinically meaningful endpoint. So actually, we wouldn't actually then need to see that clinical endpoint because we're so confident that the validated surrogate endpoint can be substituted for it. And this would have the advantage of reducing study duration. And this needs to be based on evidence that a certain effect on the biomarker would reliably predict, a clinically important effect on that clinically meaningful endpoint that we're substituting. So ideally, you'd need evidence from trials that could give an estimate of the effect of the intervention on both the clinically meaningful endpoint and the biomarker. And ideally, you would need sort of new evidence for each disease setting and different class of intervention because there could be a risk of off-target effects. And there have been some sort of regulatory pitfalls here. So, classic examples would be in HIV with CD4 counts, Also in diabetes and use of HbA1c with sort of new classes of therapies with off-target effects, where you then haven't got that clinically relevant benefit of reduction in cardiovascular disease. So obviously coming to surrogate endpoints where there is less evidence of the type that I've been talking about. So we would accept a surrogate endpoint that reasonably predicts clinical benefit in life-threatening or serious debilitating conditions where there is very high unmet need, and we would decide this on a case-by-case basis. And the potential regulatory routes would be conditional marketing authorization or exceptional circumstances, or could be the early access to medicine scheme if that was feasible. I mean, there does need to be a degree of caution, of course, for surrogates where there is only a natural history correlation, because as Emmely was explaining, it may not be on the causal pathway, so the intervention is influencing the biomarker, but in the end, it actually does not benefit the patient. I haven't got a slide on this, but I just wanted to say a little bit about placebo-controlled studies in rare diseases such as MPS. So we are in the final stages of drafting a guidance that is actually on use of historical controls of real-world evidence. So we will be publishing that, we hope, in the next few weeks. But I think from what I've heard today, this would be, a classic sort of setting for thinking about an external control such as that. So just wanted to mention that. Okay, so moving on to biomarker assay validation, and Peter was obviously talking about this. This is clearly really important when you're going to use a surrogate biomarker as a primary endpoint. And from a good clinical practice point of view, you do need to be generating reliable data if regulatory decision-making is based on the data. And actually, if anything, it's even more important in rare disease, where there are the precious trial subjects with less data, so you want to be really sure that your biomarker measurements are valid and can be used for decision making. And there is some guidance on that. Actually comes from EMA, but we adopted that at the time of EU exit, and we still refer to that. So obviously exploratory biomarker endpoints that are not a primary or key secondary to investigate mechanism of action, we're not going to be digging into the assay validation of those. But obviously, if in the end you wanted to use that as evidence to support a future validated endpoint, then obviously it is worth thinking about the reliability of that data and making sure your assays can be as well validated as possible. Okay, so just a note about clinical trial authorization. Obviously, one of our functions is to review clinical trials that are being conducted in the UK in line with our regulations, and that is to protect the rights, safety, and well-being of the study participants. So we are ensuring that the benefit-risk balance is favourable for the trial participants, and that the trial design is scientifically valid. But when we approve a clinical trial, we're not evaluating the suitability of that trial to support a licensing decision, and we're not really seeking to optimize your protocol. So really, it doesn't in itself imply validation of the surrogate endpoint. Okay, so really, I guess, the main message here is that there are lots of opportunities for you to come and talk to us at an early stage of development. So you can come and obtain specific scientific and regulatory advice on the development of a single medicinal product. You can ask for advice on the clinical trial authorization aspects if you want to conduct the trial in the UK or have a UK site. Also, you can come to us for advice on the marketing authorization aspect. So could these data support a license? Of course, you can come at the same time for both, and you indicate that when you apply, and we make sure that we get the right experts in the room, which these days is usually a virtual room. You can also come for broader scope advice. So through the same process, using that link, you can come and talk about the acceptability of a surrogate endpoint across a range of products, for example. And we also offer a fee waiver for SMEs for scientific advice. We also have our innovative licensing and access pathway, which has recently been relaunched, and we're really focusing on accelerating the time to patient care for the truly transformative new medicines. And this pathway is open to commercial and non-commercial developers, so academics as well as commercial developers. And it allows collaboration with us, the regulator, but also other parts of the healthcare system, and also, very importantly, HTA, so health technology assessment bodies in the UK, such as NICE and SMC. Okay, so just to summarise, in the context of a life-threatening or seriously debilitating rare disease, such as MPS, we may accept a surrogate biomarker endpoint if it can be demonstrated that it reasonably predicts clinical benefit. Biomarker assay validation is really important. As I've said, a UK clinical trial authorisation does not in itself imply that the surrogate endpoint is validated or could be used for a licensing application. And developers of medicines for rare disease, so both commercial and non-commercial, are encouraged to engage with us at an early stage. Thank you very much.
Thank you so much, Shirley. You can imagine my pile of questions for you both is rather large. So I'm going to try and combine a couple of them because they have a similar theme associated with them. So for EMA colleagues, there's a couple of microphones floating around, hopefully. Perfect. So we've talked about a 2-year follow-up might be a short time for a clinical endpoint. If that time was 4 or 5 years, do we still need a placebo-controlled trial, and is it ethical? And I think the reason we want to talk about that is that these are ultra-rare diseases. As I think Dr. Muenzer talked about, the number of patients is relatively small. We also heard earlier on this morning that sometimes you choose a small batch of patients to undertake the trial in. So can we maybe both Emmely and Shirley maybe comment on that and how what your thoughts are around placebo-controlled trials and that kind of follow-up time frame?
Shall I start? I think with respect to the external control question, there are certain circumstances where it would be perfectly valid to have an external control arm. And one of the situations where that's most easiest is when in the natural history there's absolutely no chance whatsoever that a certain point would be achieved. That's, I think, the most simple example where it would be perfectly valid to go on with a natural history comparator. But there are, of course, a lot of gray areas where it's very difficult. And I completely agree that probably 2 years is not sufficient to have a very compelling, statistically significant effect on a clinical endpoint. But as I said, consistent trends towards improvement or towards stabilization, I didn't say that, but I think from regulatory perspective, stabilization of a disease can certainly be very clinically meaningful. So it does not always have to be improvement. Taken together, all these aspects, yeah. I also have with me Jan, who's like the collective memory of our agency. I don't know if you want to add anything to this.
No. No, not to add, but we have, the thing I want to say is that we have experiences with a lot of companies. I'm sorry. To have a lot of companies that try something and then it doesn't work out. And for the surrogate endpoints, I've learned in my years that they are not very well established. We have established endpoints, of course. For example, the viral load in HIV positive patients, because otherwise you are tend to get AIDS and you die. That's a very good surrogate endpoint. But for the MPS, I think we have a lot of biomarkers. One is more important than the other. I like to see them. In the assessment, we like to see some hint of a clinical effect. And that is not only in the cognitive function, but an improvement in activities of daily life or communication or something is as helpful as it is. And in the end, we can consider you as an exceptional circumstance, in exceptional circumstance, and you come back every year, convince us that nothing has changed or improved, or you have your conditional approval, and then we make an we come to a condition, and we will see after that condition if you can demonstrate after 5, 6, or maybe 10 years that indeed the cognitive is stable or improved in some patients, because I think that most of the enzymes is a preventive treatment and not a curative treatment. And that's, I think, I can say.
Thanks so much, Jan. Shirley, I don't know where... Sorry.
Can I just respond to that in terms of one of our challenges in the MPS community is these placebo-controlled trials that have to be longer than 2 years, and therefore for a progressive disorder where you're actually harming the control group. And that's a challenge we have, and how from a regulatory perspective can you sort of...
I can give you my personal view here. I think it's unethical. Don't do that.
What do you do then?
Why?
How do you get the drug approved?
No, when you go to see, how many times, you are the expert, how many times do you need to see some differences in activities of daily life, in cognition, speech, so on and so forth? I think 1 year, maybe 2. And then you see some deviation between them.
No, but the problem is we don't have good validated markers for speech and things like that. We have cognitive testing.
You have your Bailey score. If you see some changes in your Bailey score, beneficial for your treated patients, I would like to come to us. And then we make some appointments, or maybe we make some conditions that have to be fulfilled, and you are going to the market. Don't wait 2 or 5 or 10 years. Then we have always, it is all the evidence. We take all the evidence in our decision. You can show that you are better than natural history. You can show that your patients are changing more to the normal. That kind of information is very important for us. And when you have a patient group after 2 years showing a deviation away from the natural history towards the normal development, and then you have a placebo-controlled trial. The placebo-controlled trial I need at least for your pharmacodynamic endpoint, for your biomarker, because your biomarker must be better than placebo group. So then we have 6 months of data at least.
In the MPSs, we clearly have a well-described biomarker that we know if we can normalize that or dramatically reduce it, those patients' courses change. I mean, it's the data...
All right, but in that case, it's very simple. Give me the information, give me the correlation, and convince me that indeed your biomarker is predictive for the outcome.
When would you like me to show up in your office to convince you?
I'm gonna because this is really interesting, and I've literally got loads of questions. I'm also conscious that Peter is still awake. So Peter, may I bring you in on this conversation around
Sure.
the kind of the
Sure.
length, duration of trials and the ethical challenges?
No, and I by the way, we started reasonably early in the morning, but it actually wasn't all that early in the morning. Remember, this is actually the time period where we're only 5 hours off with Amsterdam, so not too bad. The issue for us, I think, is that I guess from my perspective, when you have rare diseases that affect small numbers of individuals, what do we want to do? Do we want alpha error or beta error? Do we want to withhold a treatment that could be potentially beneficial, or do we give something that might not, work? And I think the idea of using a biomarker here to make a reasoned bet, and then following that up with trials that are conducted over the course of time, you can look over the course of years when it will become absolutely clear in an individual without a randomized controlled trial that they are not on the natural history curve, right? If the product is working. Now, the truth is, in gene therapy, we're very reassured about doing that, because in general, any product that we want to see developed is going to have a large enough effect that it should be able to meet that criteria. I think one of the problems that we have as regulators, and I have this with some of my colleagues who regulate small molecules. Small molecules often have small effects, and so they're used in diseases where it's very hard to understand what's going on when you use them. A good example is in non-genetically caused amyotrophic lateral sclerosis, where we don't understand the pathogenesis disease, and you use a small molecule, you don't actually really understand how it's going to work. Very hard to use biomarkers in that setting. That's quite different from the setting that we're talking about today, where there is a totality of evidence here. And I think, that it's epitomized in Dr. Muenzer's top 10 reasons for why you can accept a biomarker here of heparan sulfate that says, hey, reasonable thing to do. And the idea here is that if you use this as a biomarker, you potentially have a trial of a year's time. Let's just hypothetical, this is just a hypothetical 1-year trial that can get you to an endpoint for an approval. And then you can then conduct follow-up trials 3 to 5 years to turn that in from an accelerated approval in our world to a traditional or full approval. What have you gained by doing that approach? You've gained potentially preventing anywhere from 2 to 4 years of further decline in a group of children that will otherwise not have something. And what have you perhaps lost? Well, yep, you don't have the certainty. And yes, if it turns out that the clinical data never, confirm out, well, you've treated some children then, I guess, with an ineffective therapy. But the chance of that, based on everything that we're looking at, I think is reasonably low. And so we use a saying, I think, that has come to resonate with me, which is perfect is the enemy of good. This is the best we can do sometimes, and so we work with it.
Thank you so much, Peter. And I think in my experience, the rare disease community is much more open to some of these risks. It's great to see that the regulators are starting to be more open. Shirley, maybe I can ask you.
Could I just add something? It may be because we live in a slightly different universe here in the United States now. I didn't say that. But what I worry about is that if we aren't leaning into this, we will be in a place where people will try things or may be able to try things without us gathering data in an organized fashion. And to me, that's a much worse situation to be in. I'd much rather have people on clinical trials, people receiving products that we regulate and are able to keep tabs of than people just getting things that we don't have much control over.
Thanks, Peter.
Yeah,
Susan.
and Sheila, if I could just put an underline and emphasize that, right? Like part of the reason we have different regulators around the world is that we do have different benefit risk assessments around the world and that are played out in different statutory structures that global regulators have to implement. And part of it is, as Dr. Marks just acknowledged, you know, in the United States, we're leaning towards a far greater access to investigational product, and so that changes the calculation a bit. I can't speak to how that exists in other countries, but there is a variation in the statutory structure, that you each work under, and a difference in that benefit risk calculation, and it's not there to create barriers, but it does exist. And so then I think the conversation becomes, all right, so how do we navigate that? What is it that you need to see, and what are the opportunities for natural history or some other piece? So just as a former regulator, I wanted to observe that.
No, it's a really good observation. Thank you. Shirley, so we've got a question here around, it's a similar theme around the irreversible neurodegenerative damage that these children get very early. And how do we kind of create the right environment for a good trial to be undertaken to confirm and reassure regulators? Do we have any examples at the MHRA where surrogate biomarkers might be used or have been used that you've been able to kind of maybe share with the audience?
Yeah, so actually in the area of primary biliary cirrhosis actually is an area, obviously we're also looking at the new evidence that's coming through for some of those products, and it's not the only product which we accepted a biochemical endpoint for, so obviously that's an area that we're looking at. I guess some of the best examples are in oncology, probably, where, okay, it's not biochemical, but, for years we have used tumour response really as a it's not a validated surrogate endpoint, but it is widely used to support conditional marketing authorizations. And just wanted to say, really, obviously, we're not making any decisions today, and I'm not able to give an MHRA position on MPS. But, from what I've heard today, there's a real body of evidence here that I think goes a little bit beyond some of the arguments that we have seen before in our scientific advice interactions for other sort of surrogate biochemical endpoints. And there may well be enough evidence here to convince us to give a conditional marketing authorization based on the heparan sulfate with confirmatory data as a post-approval commitment. So I think I would sort of encourage the developers to come and talk to us and present their evidence on this to us.
Perfect. Well, you've taken my kind of parting shot away from me, which is what I was going to ask. But one of the questions that we've also got, and this is probably for all three regulators. So, Peter, may I start with you? In terms of rare disease and clinical trials, they're global, and obviously they try and capture as many patients across the global kind of geography as possible. But it can be difficult to find laboratories that are compliant to deliver the validated biomarker assays. Does the FDA, MHRA, and EMA think that they should allow a validated assay to be delivered from I think it says uncompliant or incompliant laboratory? And if I've got that wrong, please, whoever said that, please correct me.
So this is Peter. I think we would call it a non-compliant, perhaps. But I think what we would probably ask is that if a program needed to start with an assay that was being performed in a non-compliant laboratory, which we would consider the equivalent of a research assay, and a research assay is if to the extent that it can also be validated, is acceptable to start with. What we would probably do in a small population is ask for a plan for how it would move to a validated assay that could be used in a compliant manner. In other words, we would not want to stall development, provided we felt that there was an assay that had reasonable accuracy and precision. We believe that you can have development at the same time of having the assay mature while the clinical trial is going on, as long as we know that the assay is sound, right? I mean, we can't, there has to be some data to say that there's reproducibility and accuracy. But in some cases, the non-compliance has to do with technical issues of laboratory certifications rather than actual scientific issues with the assay. So I think we're trying to be more flexible in this, especially in this rare disease area where it's sometimes very hard to the cost of developing, for instance, a diagnostic that's fully validated that's conducted, and it can be millions of dollars, and in some cases that's more than the market for these products.
Great, thank you, Peter. Shirley, Emmely, any alternative views?
Yeah, no, I mean, I pretty much agree with Peter. I mean, we wouldn't obviously, obviously to try and prevent this happening is better, but these things happen, and obviously, we would sort of work with the applicant to see if we could sort of salvage the situation. I suppose I'm thinking that I understand it is possible to sort of save samples. So obviously one approach is then to have those samples later on sort of, looked at again in a central lab to perhaps provide that additional reassurance, perhaps as a post-approval commitment even. But yeah, of course, if we are using this as a primary endpoint and we're going to make a regulatory decision on it, then as I said in my presentation, this is really a sort of key aspect. But again, yes, there is obviously the commercial aspect as well that we don't want to sink development because of the costs of the assay.
Thank you. Emmely?
No, I can echo that, and indeed it depends whether it's an exploratory endpoint or the primary endpoint. But for the rest, I agree. Yeah.
So there are loads more, but I'm trying to consolidate them into as many shorter kind of concise questions as possible. And we've only got a few more minutes till the next session. So this kind of says one for EMA, but I also think the other regulators could come in. So Peter, please feel free if you feel there is anything. What would be the proposed alternative to biomarkers and conditional approval, particularly in the case of Sanfilippo? Noting that there's a lot of heterogeneity associated with it, and a standard trial will take more than 5 years, and of course, it's a fast declining condition. So, Emmely, I don't know whether you wanted to...
Yeah, well, I think in the earlier session this afternoon, we have seen that companies are actually able to compile quite complete data. So it's not completely impossible to get your data. So I think that's actually quite an encouraging session. And there are excellent publications out there describing what a surrogate endpoint, what a biomarker should be in order to function as a surrogate endpoint. Put it across, like measure your data against this information, make your case with us, and then we will assess it. And that's, of course, a reactive process. So I think it's very important what we've seen today, that companies and academia are working together, presenting maybe a collective case instead of all the companies presenting their own case. I think there's really much to gain if all the data that is out there is put together and a collective case is made, and then we can obviously assess. So I would not close that door. It's just that I would be very, very happy if I would, as an assessor, would have the ammunition to go to CHMP and say, see, they have shown that this can actually be used.
Fantastic.
I think one of the challenges is for the data you heard, This was a trial that basically was taken by one company, and then they went out of business because they couldn't do the long-term follow-up required. This was a gene therapy trial. I think for ERT, it's more challenging where, for gene therapy, you do it once, and you can follow those patients forever and know what's happened. But for an ERT trial, if you stop it, those patients are going to deteriorate. And so that's the challenge in terms of that.
I know that, if I may react to that, what I've seen, phase 2 studies, phase 1-2 studies, with, for me, reasonable impressive results. You start with a phase 3 study, you still have your patients treated in phase 2. Come after 1 year when you have demonstrated at least some benefit based on the biomarker, benefit based on a little bit of your other markers, your clinical endpoints, combine that with your phase 1-2 studies, and then we can look at it.
One of the challenges is for these MPS disorders, they're so heterogeneous that it's very challenging to get a unique population that's very concise. And so we see patients who are 2 to 6 years of age, and they very, very different clinically.
Yeah, but the problem we have is we don't have your immense experience.
But I'm happy to
That's
share
my
that.
Oh yes, we talk about that later. But the problem is that we need some real evidence in the sense that when I look at the phase 1-2 studies and you see that these patients are not deteriorating as expected, and I think Fritz has made some natural history studies about that. And when you can show that they are moving towards normal development, I think that is a reason to come. When you're coming for us for scientific advice, we play safe. We say, come with a placebo-controlled study and so on and so forth. I remember a case when I was assessing progeria, in which all patients worldwide were included, and we had a lot of discussion about the beneficial effect. That was a bad register, a bad study, but in the end, they got their authorization, except in circumstances, of course, but they have their authorization because we are open to discuss that and we are not blind for the patient's benefit. But there is a difference between to be safe in a scientific advice. On national level, we are more loose. We can interact a little bit more. When I'm going to foreign scientific advice and the EMA, I play safe because companies, I've learned companies will hold you on your advice. So,
No, I think for me is the key is that what you heard today, the scientific knowledge has been there for 10 years for what we talked about today in terms of heparin sulfate as being a biomarker that's reasonably likely to predict clinical benefit. We're never going to get a pure qualified surrogate because of the nature of the population. There's so few. We have to get a few studies approved, so it's like, it's just a chicken and the egg issue. But the scientific data is there. What you heard today is really, only a portion of what's out there in terms of showing you how that CSF heparin sulfate clearly will make a difference in these patients' life if we reduce it. If we don't reduce it, we know they're going to continue on.
I'm going to have to stop the panel there.
Well, no, just one thing that but you said this was only a portion. I personally had hoped to see more of the evidence on screen, and now it was a lot of statements from you, for example, and it's not that I don't believe you or don't want to rely on your expertise, but it show us the data. And in the end, I think also something that I find sometimes difficult is that whether or not something's ethical is also, there's a lot of aspects playing in on what is ethical. And I agree, doing a placebo-controlled trial for 5 years is not ethical. I think that's what we can agree on. But also, I personally feel it's not ethical to prescribe drugs to a patient of which you do not know whether it works. And gene therapy, it has large promise, but it's not risk-free. And we have excellent patient representation also in the Committee of Advanced Therapies, and I've talked a lot with those patient representatives, stating that, you know, that's maybe for less severe life-threatening diseases. If you are stable on ERT, she said, I would never give my child a gene therapy, while maybe another parent makes a different decision. So that's also something that I find a bit difficult in the discussion. But yeah.
No, I think these discussions are really valuable for the community. But thank you. I think we have different perspectives, and we need to continue talking.
Thank you. So I think what we're hearing from all our regulator colleagues is they are open for discussion and they're open to hear more and want to learn more. So let's find an opportunity to share some of what we know already, to open those discussions. There's obviously openness to flexibility in terms of the trials, and there are opportunities for scientific advice. And I think Jan said he's happy to be a bit more flexible if he's talking to you one-on-one. So there may be opportunities to speak to people slightly off the record and explore how we can present this case and get some progression. So thank you so much to our fantastic panel. We've got no break, but I want all of the panel speakers for the next session to come up whilst everyone's applauding this. So whilst the panel come up on stage, this is our kind of final session, and I'm sure you're all wanting to ask a million questions, but we have got some questions prepared. So I'm really excited to have some of our speakers from today join us. And I'd like to maybe start the conversation with Matthew, if I may. So it might cover some of the questions that we've had already, and there's a bit more of an exploration opportunity. So, Matthew, why do we think heparan sulfate is considered an appropriate biomarker for neuropathic metabolic disorders? Surely there are situations where heparan sulfate could come down and a patient may not respond clinically.
Sure. So, heparan sulfate accumulation in the lysosome is what causes the neuropathic disease in the MPS disorders that have that complement of disease in their phenotype. There's really no doubt about that. The issue is this is a neurodegenerative disease, and we know from all the neurodegenerative diseases that are associated with age, there is, so to speak, a point of no return. And we don't have any treatments for those because we don't understand. They don't have a simple metabolic basis. In these diseases, if you have someone with advanced neuropathic Sanfilippo who's non-ambulatory, not taking food by mouth, and you correct their metabolic defect, they're not going to get better. That's not the kind of situation we're talking about. Stabilization of individuals who are ambulatory, are taking food, that is a win. And then treating individuals who are pre-symptomatic. And that is the model of treatment. Imagine that neuropathic MPS is a slow-moving phenylketonuria, right? And how many decades have we been treating and screening for that disease and know that it works? It is that simple. We need to make sure that we prevent heparan sulfate accumulation. There is a window within which probably you get optimal treatment. We know that in MPS 1, that window is between 2 and 3 years of age for heterologous transplant. We don't know exactly where that window is, and for Sanfilippo, we know cases where kids are getting treated and are being stabilized. I mean, look at the films of Cole today and other testimonies provided. So yeah, I think there are cases where it won't, but there are the majority that will. And one thing I want to make a point on, I work a lot in public health aspects. We can't put forward proposals to screen for these disorders until we have a treatment. And we can't optimally recruit pre-symptomatic patients to prove the optimal efficacy of a treatment until we have newborn screening. Joe talked a little bit about chicken and eggs. For me, this is the killer chicken and egg, right? The minute we get a PDUFA date filing that's positive on the 18th of August, we will be filing a application to do newborn screening in MPS 3A in the states where we can. So that's an important element as well. The future really holds universal screening in countries and universal treatment for these disorders, and that's the most optimal benefit. And we have to have therapies to do that.
Thanks so much. Joe, maybe I'll come to you. I'm interested to understand how quickly you think we can observe improvements in the biomarkers like HS in CSF after treatment, and what kind of time frame should we be communicating around clinical benefit?
Well, I think we can see the reduction in biomarkers probably weeks or months. I mean, we do it. We know the liver shrinks very dramatically in the clinical trials for MPS 1 and 2. I could tell who was on treatment by just examining those patients within 2 or 3 weeks. You know, clearly within 4 to 8 weeks, we know the biomarkers are dramatically down. That's the easy part. The clinical benefit really determines that when you start, how impaired that child is will tend to depend. You know, the more impaired, it's going to take longer to show the benefits, like, for example, continuing to eat. You know, we don't have a lot of validated biomarkers. That's one of the challenges we have. You know, there's these softer things in terms of clinical outcomes should be validated, but they're not. And so it's going to take us much longer to see that clinical benefit, and that's the challenge. Our patients don't have that. You know, these placebo-controlled trials, you know, we're going to sacrifice, you know, almost a generation if we had to go continued, you know, treating with a placebo group. You know, 2 years, 1 year is clearly not enough to show the difference, and maybe 3 or 4, and that's just, to me, unethical for regulatory to say you have to put a child into parents will go into these trials because they have no other option, but it's sacrificing a generation.
Yeah, that's a really good point. So, Cara, you know, as a parent who's been advocating for so long, how do you think we should approach treating patients with these conditions where treatment isn't able to reverse the damage that's already occurred, but can improve that quality of life in the way that Joe has described and delay the progression? How do you think we should approach that?
Yeah, thank you. Well, first of all, the term quality of life, I think, is often said in a context as if it is less than any other clinical endpoint. So quality of life is composed of clinical endpoints. Okay, so it's a matter of what are your clinical endpoints. And so in a patient who is, you know, maybe not a newborn, an infant, a toddler, what are the appropriate clinical endpoints? And I think that needs to be the discussion. And they're the things that we highlighted earlier. There are a number of things, but I think the things that lend themselves most to objective measurement are things around mobility, things around oral feeding and drinking. And there are some, even if they have not been discretely collected over time, there are some binary things that clearly we can measure, like, does the child have a gastrostomy tube? Obviously, there have been significant clinical impact for a child to undergo a gastrostomy tube placement, which can tell us a lot of things. So I think there are things that even in ongoing studies that we will be able to extrapolate. It is very critical that we, yes, plan for the next generation, but let's not fool ourselves that we are discriminating against children with intellectual disabilities as if they don't deserve treatment, and they do. So we need to be embedding that in everything that we're thinking and doing around these diseases.
Thanks so much, Cara. It kind of brings, Simon, did you want to respond, or Joe, did you want to respond?
I want to respond.
Please.
I think what the totality of the data is so crucial. I think that's what Cara said. We need to look at everything and not just that endpoint with a p-value, which is challenging to get when you have small trials of 30 or 40, 50 patients. Simon.
Yeah, no, I fully agree. And I guess to come back to that point that was mentioned about that a reduction in CSF heparan sulfate does not always correlate to clinical benefit. I think I think I've said that I think what Cara teaches us is actually that there is almost always going to be clinical benefit. It's just that in certain stages of neurodegenerative disease, we can't measure that clinical benefit. So very early on in the disease, it may be that cognitive testing is good at picking up that clinical benefit and measuring it and giving us the number. Much later on in the disease, there is still likely to be significant clinical benefit. It's just, I have no idea how to measure that. And
We do have ideas about how to measure that. Yes, we definitely do. We can talk later.
And I think you're absolutely right. If I were to put my old NICE hat on when I used to evaluate therapies for rare diseases, one of the things that I used to feed back a lot to companies was it's not just about these endpoints that you're measuring, you're having to have statistical significance of and numbers and graphs. It's also about the story. What is that person living with that condition experiencing when they receive the treatment? What were they like before and what were they like after? And the Duchenne's example is a great one, where those fine motor skills that were gained from some of the therapies that are out there now are so valuable to not only the person living with Duchenne's, but the family, who can then see that child being able to feed themselves, brush their hair, brush their teeth. All of these things are really important, and I think that we need to remember that these values do translate into real life impacts. And we may not necessarily be able to put a number or a graph beside it, but actually we need to put a story behind it, which I think is really important. So Simon, We've touched on this in some of the presentations, but there are clearly some ethical and non-ethical perspectives that we need to think about here, because we have got a small population. They are, the degeneration is happening at a fast pace. How do we kind of manage that in terms of randomized studies in medical research so that we can kind of think about this progressive childhood dementia that we're trying to kind of mitigate?
yeah, so it's a very good question, and I'm glad that was brought up before, and I'm glad for the discussion that was had and the regulatory view on that. I think one of the things that's important to say that I think as clinicians, and I suspect also as patients and patient advocates, that none of us want bad trials with bad data and with bad drugs. None of us I don't want to make treatment decisions based on CSF heparan sulfate. I want to make treatment decisions based on really good, hard data, and we all know that that's best in a placebo-controlled, double-blind, randomized study. But where that is not possible, or where those attempts have led to no new therapies whatsoever, we have to accept lesser standards. And I think, for me, the best approach would be using something like an accelerated approval with a biomarker to allow some of these very small companies to keep existing and the trials to keep running. And I think we would design much better long-term trials if we weren't desperately trying to show something clinical in the first 12 or 24 months. I think our attempts to do that and to fit these really complex, difficult diseases into that model, which was made for small molecule drug development, has led to some really quite bad trials. And in the end, we've put a lot of children through some very invasive, very risky therapies, and not at the end of that had good data. And that feels to me the most unethical thing here, is that we've put hundreds of children with neuropathic MPSs through trials that have not yielded data that has allowed us to make decisions. And so the children that I diagnose in my clinic today, for the majority of these diseases, I have nothing to offer them. That doesn't mean we should all accept, terrible data. That's not what we want. So I think it's important to be able to have an open discussion with the community about what is and what isn't ethical, because those are really difficult decisions. They will vary from disease to disease and from therapeutic modality to therapeutic modality. And we have seen in the past some trials that have been approved that, in my view, were clearly unethical. They have been, those trials have, and they have recruited patients because families will have no choice but to enroll into these studies. But the decisions often about what a trial is, the ethical nature of a trial or not, is had with a regulator and a company, sometimes a small, naive company, with no offense meant, behind closed doors, rather than more broadly with the community of patients and clinicians and academics and those who've been living and working with these diseases for decades often. So that would be a way, I'm not saying that's easy, of trying to make things more ethical.
Thanks, Simon. I'm conscious of time, so Cara, I might go to you for the last question. You know, speaking on behalf of patients and families, what final words would you want people to take away from this meeting?
Well, I think I probably said a mouthful in the last, in my last response, but honestly, that these conditions need to be looked at in a very disease-specific way. We don't need to be even comparing with one another. I mean, each disease is each disease, and consider the timeline that that disease demands. And what we've shown is that the timeline that the diseases demand does not fit into the regulatory pathway as it stands, and continuing to kind of blanket recommend is leading to people not coming into the space, sponsors not coming into the space. We don't care inherently if a company does well or not. But we care if our children have access to clinical trials and eventual therapies. And so the financial health of a company being able to sustain the access for us is important. And our regulatory systems have to be able to enable that to happen. So we're all working in an ecosystem together. And patients, people living this life need to be at the table in the process. Yeah.
Thank you so much. It's been a fantastic afternoon. I have the pleasure of handing over to Kim Angel, who is going to give us some closing remarks. Did you want the microphone? You going to bring it? Okay.
Hi. So first of all, it's a great honor to be working in partnership with the MPS UK, Bob and Sophie. Thank you so much for your heavy lift in developing and executing this amazing meeting. These conversations being opened with the EMA and MHRA today are so incredibly important. They are conversations that are happening. We know that we're not walking away with any changes at this point in time, but what we're hoping to do is enable these conversations to happen, to start accelerating treatments for our children. I think a theme that's been interwoven all day long is time. These kids don't have time. These parents want more time. This morning, Cara and Kim and Mark demonstrated the very sobering effects of neuronopathic MPS on children and families. Cara also pointed out another sobering fact of how many clinical trials have ended because of the regulatory process. We just don't have time to continue this way. Joseph, you mentioned a paradigm shift. The United States, fortunately, has begun a paradigm shift, and we really hope that this will extend globally. This knows no geographic boundaries. It knows no socioeconomic boundaries. This disease is devastating, and we don't have time. So I'm very happy that the EMA and the MHRA here were here to see the science. It's there. The science is there. We're happy you're open to discussions and open to looking at all of the science. And we know that the companies are working very hard to ensure that this data can be presented to you in a way that will help accelerate pathways for these children to live lives. So on behalf of the IMPSN and also on behalf of the MPS Society UK, thank you all for your dedication to these patients. As a patient advocate, I have seen and have been a part of so many neuronopathic MPS patients' lives where you have to tell them there's nothing we can do. I would really like to see this changed as quickly as possible, and I know everyone in this room does as well. So thank you very much, and we look forward to further conversations.
Thank you so much for everybody who joined in person. It was a pleasure to moderate this session. And to those of you that are online, you can finally go to sleep if it's nighttime. And those of you that have gone and worked through the night, good luck on your office day when you get there. Thank you so much.